In this video, we can discuss about the different media or substrate which are used for the industrial fermentation process. We have already discussed the ideal structure and design of an ideal fermenter and its working. Now we can discuss about what are the different medias used for the industrial fermentation, not for the laboratory type of fermentations. So first of all, while considering the media should contain all the elements in suitable form for the synthesis of cellular substance as well as the metabolic products. While designing the several factor, designing the media, several factors must be taken into the consideration. The most important of them is the ultimate product desired in the fermentation process. Depends on the ultimate product desired in the fermentation, we have to change the media. For example, for growth linked products like primary metabolites like ethanol, citric acid, in this the product formation is directly depends on the growth of microorganism. Hence, the medium should contain all the elements which will support the growth of microorganism. Now, for the products which are not linked to the growth, that is secondary metabolites like antibiotics, alkaloids, and gibberellin, the substrate required for the product formation must also be considered in that way. In that type of product, the desired product is whether it is secondary metabolites, we don't have to consider the growth of microorganism. We have to consider it is a secondary waste product. Maybe it is the waste product of microorganism. So we have to consider the secondary metabolite like antibiotic, alkaloid, and gibberellins. Now, in laboratory, we are using pure defined chemicals for the culturing of microorganisms. But in case of industrial fermentation, undefined and complex substances are frequently used. While selecting, we have to select a substance which is cheaper in cost because we can minimize the production cost. The choice of medium is very crucial for successful product formation. It should contain abundant carbon, nitrogen, and growth factors. The media used may be synthetic or crude media. Synthetic media is the media in which all the requisite constituent in the pure form in desired proportion. But it is practically not possible. So we will use crude media. Crude media are non-synthetic media which naturally available source with an addition of requisite substance request synthetic constituent is ideal for good growth yield in the formation. The most frequently used substance with special reference to the supply of different substrate used for carbon, nitrogen, and growth factors. Now we will consider one by one what are the different substance used as a carbon source. So we are using carbon source for the purpose of prominent source of energy. Usually carbohydrate constituents are used for the most prominent source of energy. Now what are the different carbon source that we are using frequently? First one is molasses. Molasses is a byproduct of sugar industry, one of the cheapest source. Sugarcane molasses, sugar beet molasses are commonly used. It also containing nitrogen substances, vitamins, and traces of elements. Now second one is malt extract. It is an aqueous extract of malted barley which containing 80% carbohydrate like glucose, fructose, sucrose, and maltose, and 4.5% nitrogen compounds like protein, peptide, amino acid, purine, and pyrimidines. Now other carbon sources are starch, dextrin, and cellulose, which are mainly used for the production of alcohol. Then whey, it is a byproduct of dairy industry used for the production of alcohol, single cell protein, vitamin B12, lactic acid, and gibberellinic acid. Then methanol and ethanol, which are the cheapest substrates, but it can be utilized by only few bacteria, and yeast, which are used for the production of single cell protein. So these are the different carbon sources used for the production, or used for the used as an culture media or substrate for the fermentation process. Now second one is substrate which are used for the nitrogen source. Nitrogen source are of two type, whether it is inorganic or organic. Inorganic nitrogen source like ammonium salt, ammonia are the cheapest source, but not all the microorganisms are capable of utilizing them, hence it is used in very limited cases. And in case of organic nitrogen source, we can use urea, corn steep liquor, which are formed during the starch production from the corn, and which is rich in several amino acids like alanine, valine, methionine, arginine, threonine, and glutamate. Now yeast extract, another organic nitrogen source is yeast extract containing 8% nitrogen, and which are rich in amino acid, peptide, vitamins, produced from baker's yeast through autolysis at 50 to 55 degrees Celsius, or through plasmolysis at high concentration of NaCl. Now soya meals, another organic nitrogen source is soya meals uh, prepared by extracting from soybean oil, which are rich in protein and carbohydrate. And last one is peptone, protein hydrolysate, referred as peptone. Protein hydrolysate are referred as peptone, which are the main source are meat, soya meals, peanut seed, cottonseed, sunflower seed, casein, gelatin, and keratin can be hydrolyzed to prepare peptones. So these are the different nitrogen sources. So next one is the substrate which can be used as a growth factor. Usually microorganism cannot synthesize growth factor, so we have to include this in culture media. Now but in pure form it is very expensive, hence crude sources are preferred, like yeast extract is a rich source of all the growth factor. So these are all about the different culture media or substrate used for the industrial fermentation, and it may vary with the desired product of fermentation. In detailed for the production of each product, we will discuss the different specific culture media used for the that specific products. Now we can discuss the sterilization method of culture media. The basic principle of fermentation is we are using a organism, and by using that organism we are converting a crude substance to a structurally similar desired product. So we have to make sure that the culture media do not containing any type of other microorganism other than the microorganism we are using for the production of a desired product. So in order to acquire that sterility or freedom from other microorganism, we will sterilize the culture media. So the constituents of culture media, water and contribute to the contamination by vegetative cells as well as spore. So the medium should free from all the contamination before using for the fermentation, and it can be achieved by applying heat, which is the most common method, or lesser extent in by other means in case of heat labile substance by using physical method, chemical method, radiation methods. Now first one is let's discuss the physical method. It can be done by filtration, centrifugation, and adsorption, which is used for certain constituents like vitamins, blood components, antibiotics of the culture media, which are heat labile, which will be destroyed by the heat sterilization. So we will use filtration, centrifugation, and adsorption for heat labile substances. Now what are the limitations of filtration? Application of high pressure in filtration is unsuitable for the industry, and some of the media component may be lost due to the filtration. So other method of physical method is by using disinfectant, but it cannot be used frequently because it will contaminate the desired product also. And in lesser extent we can use UV rays, radiation sterilization method, by using different rays like UV rays, gamma rays, which are not commonly used for the medium sterilization because these are expensive and practically not possible inside the fermentation process or inside the plant. Now, most commonly used method of sterilization is heat sterilization method. Now, while selecting the sterilization method, heat sterilization method, we have to consider some factor which will influence the quality and quantity of the contamination, composition of the culture media, its pH and size of suspended particles. Usually vegetative cells which can be destroyed at lower temperature in short time, that is around 60 degrees Celsius in 5 to 10 minutes. But in case of spore which requires high temperature and relatively long time, that is around 80 degrees Celsius for 15 to 20 minutes. So this is a common method of sterilization by for vegetative cell. We have to heat it for 60 degrees Celsius for 5 to 10 minutes, and in case of spore we have to heat for 15 to 20 minutes at 80 degrees Celsius. So sometimes we can use Bacillus stereothermophilus, which are most heat resistant. So it is used for the sterility of the fermentation equipment and media after the sterilization to check whether it is sterile or not. Now heat sterilization can be done by two methods, by batch or continuous method. Now first one, heat sterilization by batch sterilization. The culture media subjected to the sterilization at 121 degrees Celsius in a batch volume of bioreactor. It can be done by injecting the steam into the media, which is known as direct method, or injecting steam into the interior coil, which is known as indirect method. But these have some disadvantages, like it will cause the damage to the culture media, alteration in the nutrients, change in pH and discoloration of culture media can be occur. Then high energy consumption, it may take few hours, 2 to 4 hours, for entire content of bioreactor to attain the requested temperature. That is the disadvantage of batch sterilization method. Now, so we can use second method, that is continuous sterilization method. It can be carried out for continuously, that carried by at 140 degrees Celsius for a short period of time ranging from 30 to 120 seconds. This is based on the principle that the time required for the killing of microorganism is much shorter at higher temperature, and it can be done by directly injecting the steam or by means of heat exchanger. Three type of heat exchanger are used for the continuous sterilization. The first heat exchanger raises the temperature to 90 to 180 120 degrees Celsius within 20 to 30 seconds, and the second exchanger further raises the temperature 140 degrees Celsius and maintain it for 30 to 120 seconds, and third exchanger bring down the temperature by cooling in next 20 to 30 seconds. So this is a usual procedure for the continuous sterilization process. Now, what are the advantages? About 80 to 90 percentage of energy is conserved because it will take only few seconds, like 30 to 120 seconds for the total process of continuous sterilization, and we can sterilize the culture media continuously one after one. So that is the heat sterilization process. Now we have discussed in the working of fermentation process, we have to give aeration in order to give the fresh oxygen and to remove contaminating or exhausting carbon dioxide from the fermenter. So while giving oxygen or aeration, we have to make sure that it is sterile, it is free from the contaminating microorganism. In order to that, we have to sterilize the air. The air or other gases can be sterilized by filtration method, or by heat or UV radiation, or by gas scrubbing method. The air sterilization by heat is by passing over electrically heated elements. It is quite expensive, so it is not used nowadays. The most commonly used sterilization method for the air is filtration method, which can be sterilized by using different type of filters like depth filters, which are composed of glass wool containing depth filters that particular trapped and removed. This filtration is primarily involved the physical effects like inertia, blocking, gravity, electrostatic attraction, and diffusion. Glass wool filter can be steam sterilized and reused. So that is the depth filters. Another filter which are used for the sterilization of media is membrane cartridge filter. These are removable plated membrane filter made up of cellulose esters, nylon or polysulfone. The membrane cartridge filter are smaller in size, simpler for the operation and replacement. Now what are the limitations? There is no filter which can remove the bacteriophage. So while sterilizing the air, we cannot remove bacteriophage, which is a complex virus in by using filtration method for the sterilization of air. So that is the sterilization of air. Now coming to the next part of fermentation process, we have to inoculate the fermenter with a specified microorganism. Now how we will select and how we will isolate the microorganism? So we will use natural source itself like soil, lake, liver, modes. These are the natural source for isolation of microorganism. There are millions of species of microorganism which are widely distributed in nature. Less than 1% of this have been studied till now. Now for example, if we are taking 1 gram of soil, which is a major source of microorganism, it containing 10 raised to 6 to 10 raised to 8 bacteria in 1 gram of soil, and 10 raised to 4 to 10 raised to 6 actinomycetes, and 10 raised to 2 to 10 raised to 4 fungal spores. So that much number of microorganisms are present there in 1 gram of soil. So it is a major source of microorganism. Now what are the different microorganism and the desired product or from which are the product which can be obtained? For example, from bacteria, Acetobacter aceti, that is the microorganism. By using that microorganism, we can produce acetic acid by fermentation process. Then Acetobacter woodi, again acetic acid can be produced. By using Bacillus subtilis, we can produce bacitrine. Then Bacillus brevis, gramicidin, Bacillus thuringiensis, endotoxins, Clostridium aceticum, acetic acid, methylophilus methylotrophs, glutamic acid, Pseudomonas dendrificans, vitamin B12. Now by using algae like Chlorella sorokainia, single cell protein can be obtained by fermentation process. And Spirulina maximum, again single cell protein can be produced. Now by using fungus, the major source of so many pharmaceutical products is fungi like Aspergillus niger for citric acid, Aspergillus oryzae for amylase, cellulose and single cell proteins, Candida lipolytica for production of lipase, Candida utilis for the production of single cell proteins, then penicillin chrysogenum for the penicillin, then Saccharomyces cerevisiae for production of ethanol, wine and single cell protein, Saccharomyces lipolytica for the production of citric acid, single cell proteins, Rhizopus nigricans for the production of steroids like corticosteroids, prednisolone, etc. Then Gibberellina fujikura for the production of gibberellin, then Trichoderma viride for the production of cellulase, which is an enzyme which will digest the cellulose. And then by using actinomyces like Streptomyces oreofascins for the production of tetracycline, Streptomyces griseus for the production of streptomycin, Streptomyces stradii for the production of neomycin, then Nocardia mediterrana for the production of rifamycin, then Monospora purpura for the production of gentamicin. So these are the different microorganisms and its product. which can be produced by fermentation process. Now, how can we isolate the microorganism from this technique or these sources? We can use different methods like direct sponge of soil, by soil dilution method, by diluting 1 gram of soil, and we have to make from that 1 ml to 10 ml, like that, by using fresh culture media. Then gradient plate method by using solid pour plate method or streak culture method. Then aerosol dilution method, flotation method, centrifugation method, and enrichment method. This enrichment method is an important method for the isolation of microorganism according to the nature of the microorganism. So different type of microorganism can be isolated by giving its environmental condition, like for thermophils by using high temperature, that is 40 to 120 degrees Celsius. For psychotrophs, which are the organism which will grow at low temperature, so by giving a low temperature of 5 to 150 degrees Celsius, only the organism which will grow at that temperature will be there in that product or in culture media. Then acidophiles by using low pH, halophiles high NaCl concentration, anaeros in an nitrogen atmosphere, and then actinoplanes by using pollen grain, and myxobacteria by food barks. So these are the different isolation method of microorganism. Now, uh, now how can we screen the metabolites for the isolation of microorganism? So microorganism can be tested directly for the product formation and can be isolated. For example, agar plate can be used for the screening metabolite formed from the microorganism. For example, for an inhibitory substance like antibiotic, it may produce zone of inhibition, which indicate the possible presence of an antibiotic being produced by that microorganism. So we are using a culture media, solid culture media. Into that we are giving an microorganism and we are allowing to grow for some time. So if after the growth of that microorganism, if that solid plate shows some zone of inhibition, it indicates that microorganism can produce antibiotic like substances in that culture media that we are provided. So that is the screening method. Now in case of isolation of microorganism which will produce amylase, can be identified by growing on an agar plate containing starch which is stained with iodine, so it will give a blue color. So by this method we can screen the metabolites which are produced by the microorganism. Now, screening of new metabolites for identifying the chemotherapeutically important products for the treatment of tumors, bacterial diseases, and viral diseases. Several other substances can be used. Other substance can be used. Now, how can we preserve? Now we had isolated the microorganism. Sometime our fermentation process will be after 1 month or 2 months, so we have to preserve that microorganism for that 2 months. Now what are the different methods? We have already discussed in the microbiology, pharmaceutical microbiology, what are the different methods for preservation of microorganism. In that we will use the refrigeration as well as freezing or lyophilization method, the preservation of microorganism. So that is the isolation of microorganism. Now, depending on the product, so we know we are taking on culture media and we are giving an inoculation containing a desired microorganism for a desired product. Now we are allowing to grow the microorganism at a suitable temperature and conditions. It will produce some metabolic products, or it will produce some end product which may be a primary metabolites or secondary metabolites. We have to make sure that our desired product is primary metabolites or secondary metabolite. According to that, we have to change the culture media and we have to get the more product, whether it is primary metabolites and secondary metabolites. Now let's discuss about what is primary metabolites and what is secondary metabolites. Primary metabolites are essential metabolites or which are required for the growth of microorganism. These are the metabolites which are produced by microorganism and which are very essential for the existence and the reproduction of microorganism itself. Now it is a this type of primary metabolites is characterized by the balanced growth of microorganism. Whenever the growth is balanced, then only this primary metabolites is produced. It is also known as trophophase. It occurs only when all the nutrient needed by the microorganism are provided in the media. So there are two type of primary metabolites: primary essential metabolites and primary metabolic end products. So primary essential metabolites are produced in adequate to sustain the cell growth. Example like vitamin, amino acid and nucleoside. Then primary metabolic end products like normal and traditional end product of fermentation process of primary metabolism. Example are ethanol, acetone and lactic acid. Now how can now we if the desired product is primary metabolite, we have to select a culture media to get more primary metabolite. That is our desired product. So what have to do for overproduction of primary metabolites? So by we can produce overproduce by eliminating the feedback inhibition by using oxotropic mutants, which will block in one of the step in the biosynthetic pathway concerned with the formation of primary metabolites, or by using mutant microorganism with anti-metabolite resistance which exhibit defective metabolic regulation. These are the method for the production of overproduction of primary metabolites. Now second one is secondary metabolites, also known as idiophase. As the exponential growth of microorganisms ceases, they enter into idiophase, which is characterized by the formation of secondary metabolites or idiolites. The thing is, the secondary metabolites are the product which do not required by the microorganism, but they will produce it abundantly. It do not have any role for the growth and reproduction of microorganism. It will produce abundantly. We can use that one. No need for it. Do not need for the microorganism for its growth and reproduction. Now examples are antibiotics, steroids, alkaloids, gibberellin and toxin. These are specifically produced by selected few microorganisms which are not essential for the growth and reproduction of which may perform some unknown functions. Then environmental factors may influence the production of secondary metabolites. Some organisms produce a group of chemicals. Biosynthetic pathway of secondary metabolites are not clear. Now, how can we overproduce the secondary metabolites? Sometimes we required antibiotic like penicillin and steroids like corticosteroid, alkaloids, gibberellin, toxin. These products have to be overproduced for our pharmaceutical benefits or for the more product. So it can be overproduced by induction by adding methionine, which will induce certain enzyme and enhance the production of cephalosporin. So for the production of cephalosporin antibiotic, by adding methionine, we can induce the certain enzyme and enhance the production of cephalosporin. As well as tryptophan, which can be regulate the ergot alkaloid biosynthesis. Now second one is end product regulation. Sometimes the end product inhibit the growth of its own biosynthesis. Example, penicillin, streptomycin, puromycin, chloramphenicol. So by selecting a mutant which is less resistant to end product, which can increases the secondary end products. So these are the method for the overproduction of secondary metabolites. Then next method is catabolite regulation by a key enzyme involved in the catabolic pathway is inactivated, inhibited or repressed by adding a commonly used substance. It is achieved by using carbon or nitrogen sources. Glucose found to inhibit the production of several antibiotics like penicillin, streptomycin, bacitracin, chloramphenicol and puromycin. So that is the catabolite regulation. By using a commonly used substance, we are inhibiting or inactivating a catabolic pathway by using a commonly used substance like glucose, which can be used to suppress or production of several antibiotics like penicillin, streptomycin, bacitracin, chloramphenicol, and fluoromycin. Then phosphate regulation. Inorganic phosphate is required for the growth and multiplication. By increasing the concentration, increases the production of secondary metabolites. So these are all about different media used and its sterilization method. Now how we will sterilize the air, and what are the different metabolic end products, and how we will overproduce the metabolic end products. So that is all about this session. Thank you for watching this video.